So very good morning to all. I welcome all the faculty members, young faculty members to utilize this session for this is the fifth day of the program and final day. And on behalf of Faculty of Pharmacy, BIHER, and as well as our Dean initiation, we initiated this FDP program and it is running smoothly for the day 5. And today the resource person, Dr. R. Venkatesha Perumal, Professor, Qasim University and Kingdom of Saudi Arabia. And the next 1 hour session, it will give the immense pleasure to the young buddies to know about the various strategies in the drug discovery, like innovative strategies in the drug discovery, exploring their structure-based ligand and based on the approaches. This is the one of the session. It will be very useful for the young faculties as well as researchers, PhD scholars to initiate their research works in the various fields. This FDP already, I said the objective is to enhance the research skills and to identify the innovation in the pharmacy, to also know about the interdisciplinary approaches and integrating with the nanotechnology, bioinformatics, biotechnology with the industrial approach, as well as collaboration with the industry, academia, as well as to know about the various regulatory and ethical consideration. So in this regard, we have discussed the various chapters for each and every day. And I request Lata Madam to give the intro speech about Venkatesh Perumal sir.
Good morning, sir. Very good morning and a warm welcome to all my dear faculty members, colleagues, honorable guests and participants to the Faculty Development Program conducted by Department of Pharmacy, BIHER. Myself is Lata, Assistant Professor, Department of Pharmacy, BIHER. It is our pleasure to introduce one of our distinguished speaker, Dr. R. Venkatesha Perumal, who holds a PhD from BITS Pilani and has been recognized as a Senior Research Fellow by CSIR, New Delhi. Currently, he serves as a professor at Qasim University in Saudi Arabia. With a profound interest in silico molecular docking, pharmacokinetic studies, and DFT studies, he is a leading cutting-edge research in the green synthesis of flaxseed nanoparticles and their potential effect on diabetes mellitus. Dr. R. Venkatesha Perumal has an impressive record of 62 publications and H index of 18 and 948 citations. He is also guiding 3 PhD students and serves as a reviewer for prominent journals including Bioorganic Medicinal Chemistry Elsevier and Archive de Pharmacy +1. We are honored to have Dr. R. Venkatesha Perumal with us and look forward to gaining insights from his extensive expertise and experience. Once again, welcome to the Faculty Development Program. We hope this program will be a valuable experience for all. Thank you.
I want to talk a few words about Venkatesh Perumal. So he is my very close friend from BPharm, and he did his PG in CL Mehta College of Pharmacy. So just few months he worked once again in and around Chennai, his native, and he went to BITS Pilani. So he is one of the first batch bright person in our college. It's a proud moment for us also to invite him, and we joined in the platform after long years. In fact, we don't have time also to talk over. Somewhat we are talking somewhere and all. So I am very much happy as a friend, as a colleague. So as we together roam, so we are here sitting together. So he is from Saudi and I am in Chennai to share the moment. So, and I once again thank my staff members. So as these 5 days went so much excellent. So people all over the world, they have shared their memories. So they have utilized the EFDP program, first of its kind, from our Faculty of Pharmacy, Bharat Institute of Higher Education and Research. So thank you very much, Venkatesh Perumal, for being here for us. And once again, the session is yours. Thank you.
Good morning, everybody. First, I thank
Good morning, sir.
everybody, sir, Lata Madam, And I have to say about Dr. Srinivasan. So we had a very wonderful college days. So after that, after the college days, okay, actually we are in constant touch in various conversation. So I really thanks to the dean, pharmacy, and also he is the who initiated this faculty development program. And I have to say some few words about another colleague in your colleagues, is Jiran Kadi. Yeah, he is, of course, she was the topper during our college days. Actually, she was the role model, I can say. So okay, we used to take her notes. We used to take her notes for the exams. We used to copy her notes. Okay, so I remember the handwriting. I can't forget. Okay, the handwriting was so okay, like beautiful. So with this, I'm very happy to see you again in this platform. Okay, so with this, I'm just starting my today's lecture. I hope my voice is clear.
100%.
Yeah, good. So today I'm just going to start about the innovative strategies in drug discovery, exploring structure-based and ligand-based approaches. In this, what I'm going to discuss is first I will just discuss about the drug discovery part. Then I will move to the structure-based. What are the aspects are there in the structure-based, and then I'll move to the ligand-based approaches. I hope all of you have some idea about this topic. Okay, maybe so those who are already having idea, maybe you can take it as a refreshing platform. Okay, so some things, I think students also, I came to know some students also, they are present online. So I think for them also, I think it will be useful. Of course, this is one of the recent trend in drug design. So with this, I'll move to the topic. As you all know that initially drugs were obtained through the natural sources. During our BPharm days, okay, we are used to study like pharmacognosy. What are the various drugs isolated from the plant products? Okay. So initially we use the drugs mostly based on the plant, that means natural based plant drug products, mainly plants. So after that, there was some accidental discovery also was there. Like everybody familiar with the penicillin, Alexander Fleming. He discovered the penicillin through accidental approach. So in our time we used to call serendipity. That means by chance, drug discovery by chance. So this is one of the classical example, penicillin G. After that, I think the well-known drug sildenafil citrate, that is the Viagra, that was also an accidental discovery, because Pfizer initially they were developed the drug for hypertension. During the clinical trials, they came to know rather than hypertension, it has like phosphodiesterase inhibitor. So that was the accidental discovery. So because of that, the Pfizer company, I think you may be knowing, you may be aware that though they have made a huge profit from the sildenafil citrate. Actually, this is the timeline of the drug discovery. It starts from the 1990s, okay, like herbal drugs, and then accidental discoveries. Slowly, okay, then vitamins, vaccines have emerged. In 1940s, it was the antibiotic era. So during 1940s only, this R&D was started coming up. In 1970s, the biotechnology, information technology also emerged in the field of the drug discovery. In 1960, there was a breakthrough in the etiology. In 1950s, the new technology has emerged, the discovery of the DNA. In 1980s, like commercialization of the drug discovery, and of course the combinatorial chemistry. This term also it is okay, a very important one, and it is also a recent trend. 1990s, robotics and automation has come. So from 1990s. Okay, so we are developing so much. Of course, as you are all aware that nowadays artificial intelligence also plays a role in the discovery. So with this timeline, I will move to the next one. So this is the picture of the traditional drug discovery process. So initially, even I remember during my PG during the CL Bade method, I think in 1999. Yeah, it was maybe informed during 99. At that time, they used to synthesize the number of molecules and then randomly go for the pharmacological activities without like much logic. So what are the facilities available in a particular college? For example, analytic, anti-inflammatory, various antibacterial studies. So without much logic, we are randomly screening the molecules. Okay, so it can be either from the synthetic or natural compounds. So that's what happened before 2000. We synthesize the molecules and go for the preclinical testing. That means in the animal studies. So that was happened. So first we'll synthesize the molecule, then go for the preclinical trials. That is in the small animal models, and then those molecules which are prominent in the preclinical trials, we'll take it to the clinical trials. And then those molecules which are which causes the clinical trials, then it can be entered into the market. So this is what the traditional drug discovery. So now what we are doing is the in first step itself, we are just making a target selection. What target I'm going to work? That's more important. So this target identification, and then we have to verify the target, whether that target is really involved in a particular disease process, and then lead identification. So once we select the target, then our role is to generate the lead. Lead in sense, I can say it is a drug, right? A drug molecule or any chemical molecule. So you know that all drugs are chemicals, but not all chemicals are drugs. I think you are well versed about that. So once we have the target, so based on the target, we can develop the molecules. So there will be developing a number of molecules. From the number of molecules, we have to find the lead. Okay, which is lead in sense, which is most prominent among these series of compounds, and then lead optimization. Okay, after lead identification, we have to optimize the lead. For example, you may be knowing that suppose if a molecule shows a good efficacy in the pharmacological activity, so it cannot be successful drug. Why? Because it has to fulfill the pharmacokinetic parameters also. Suppose there are two things. One is pharmacodynamic, right? That is efficacy, and then pharmacokinetics. All the drugs fulfill both the criteria, pharmacodynamic as well as the pharmacokinetics. Suppose the drug is having very good efficacy, but if it fails in the pharmacokinetic parameters, suppose absorption, distribution, metabolism, excretion, any of the pharmacokinetic profile, if it is not doing good, so then it cannot be a successful drug, whatever the efficacy. Suppose even toxicity also plays a role. That's what we called ADME toxicity. We called it a bad bet. Okay, so once we do the lead optimization, then we can go to the next one. We can go to the synthesize the molecules, then we can go for preclinical trials. So in the modern drug discovery, what we are doing is we are minimizing the number of molecules to be synthesized. So target selection you are doing, lead identification, lead optimization. In this process, that all these things we will be doing in silico methods. So during this process, okay, we will minimize the number of molecules to be synthesized the molecules. Which molecule is showing the most prominent in the pharmacodynamic as well as pharmacokinetics. They are both the parameters we can assess in the using in silico methods. So once we identify the lead molecules, only those molecules take it to the lab, synthesize it, and then we can go for the in vitro, in vivo models. In the clinical trials. Okay, and then suppose the molecules showing good pharmacodynamic as well as pharmacokinetic profile, then those molecules we can take it to the clinical trials. Then it can be come to the market. So this is what the difference between the traditional drug discovery and modern drug discovery. Because nowadays the pharmaceutical business is very tedious process, not like any other product. Because what the reason is, it is very time consuming. Okay? If you want to bring a single molecule to the market, it takes around 10 to 15 years. Suppose if you start a project today, now it is 2024, so you'll be launching your product by 2040. So that much year, okay, that much tedious, okay, evaluation process, okay, that has to be taken care. Whereas adhesives, for example, if you want to make it like any glue or adhesives, the chemist can develop the product within 3 months. But in the case of a drug, it takes 10 to 15 years. What is the reason? Because it has to go through the rigorous testing. For example, first we have to test it in the animal, in vitro studies. In animal, both you have to finalize it. That means both pharmacodynamic and pharmacokinetics. Okay. After preclinical trials, take it to the clinical trials, human, and then scale up approvals. Approvals also it is here because of the documentation, for regulatory affairs. So it's also one of the tedious procedures. So you have to document each and every step, okay, and then everything has to be documented, and it has to submit to the regulatory bodies for getting the approval. So that is the reason it takes huge time. So as already told you, it is a very complex procedure, lengthy, and moreover it is expensive one. So R&D is very expensive one. That is the reason most of the companies, they will not be having R&D. What is the reason? Setting up the R&D, so it will be a very costly affair. So that is the reason most of the small companies, they cannot afford to establish the R&D. So, for example, if you have like 10,000 to 20,000 molecules initially, in the later stage, each stage, it will be filtered. Suppose for example, in preclinical studies, okay, some of the molecules will be eliminated. After preclinical studies, in human, that means clinical trials, you may be knowing that. Like, it actually mostly, in generally we speak, it is happening in 3 phases. 3 phases, each phase, some molecules will be filtered out. So at last maybe, okay, we may be getting one compound to the market for the phase 4 studies. Sometimes, even one molecule may not enter into the market. That is also possible. That means the total project it will be failed. So that's the reason it is very time consuming, as well as it requires a lot of investment. So I'll just give you an overall picture on how this drug discovery process is taking place. The first step is that we have to identify the disease. What is the disease? And after identifying disease, what is the mechanism for that? What are the physiological functions? Okay, what physiological function is malfunction? Okay, Okay, what the receptors are involved, or any biochemical parameters are involved in that. So we have to identify the target. After identifying the target, isolate the protein for a particular target. So we know that, okay, isolate the protein, okay, establish the structure. I will tell you how to establish the structure in the coming slides. Okay, isolate the protein and then establish the three-dimensional shape of the structure. Once we have the structure of the target, then they can design the molecule for the particular target. Okay, so by using in silico methods. So you can in silico methods, as I already told you, we can do both pharmacodynamic and pharmacokinetic studies. We can do it. There are so many like online tools are available, and commercial software is also available for doing pharmacokinetic and pharmacodynamic studies. Okay. Once we do the in silico studies, suppose if you are having a prominent result in the in silico studies, okay, and then you can take only those molecules to the synthesizing. Synthesizing, you can suppose if you have a number of molecules to be synthesized, you can use combinatorial chemistry. So combinatorial chemistry, okay, it is so it's like a chemical library, you can say. You can at the same time you can synthesize like thousands of molecules at a time. So that is the combinatorial chemistry. I am not going in detail into the combinatorial chemistry. So for what do you call that? For optimizing the structure of the molecules, two things will be very much useful. One is the molecular modeling, both like in vitro and in silico ADME models. So both will be helpful to get a permanent molecules before synthesizing. And then, then the rest of the process is same, preclinical testing. Once the preclinical testing is satisfied, then we can file the IND, that is investigational new drug approval. Suppose in case of US it is the FDA, so you have to file the IND to the regulatory bodies. Once we get the approval, then we can go for clinical phase, phase 1, phase 2, and phase 3. After phase 3, based on the results, based on the data, we can file the new drug approval, same authority, same regulatory authority. For example, in US FDA. Okay, after getting the approval, the drug can be sent to the market. So this is the overall picture how the drug discovery process is happening. Okay, so then I will move to the biological target. I'm just in many times I just mentioned like target. Please excuse me. My language is clear, my voice is clear. Yeah, okay, fine. So then I will move to the biological target. So mostly when we say target, mostly we consider a protein molecule. As we all know that protein molecules, okay, it can be either receptor, mostly receptor or enzyme. These two things we are going to consider, receptor or enzyme. Both, when you say receptor or enzyme, it's very obvious that it's made up of various amino acids. Then what is the drug design? So drug design is finding a new medication based on the knowledge of biological target and already existing molecules. So this is the drug design. Drug design is we are developing a molecule based on the biological target. That is one aspect. The another aspect is based on the already existing molecule for the particular target. By two way you can develop the new molecules. So the molecules, whatever we are developing, that should be in complementary in shape and charge to the target. That is one of the basic things. That should be in complementary in shape. So that only it can be easily bind, right? And also in complementary in shape. complementary in charge. Okay, so only shape and charge both are important. So that only if the target on the drug is not in complementary in shape, then the binding will not be will not be take place. So you can see here. Okay, so this is the target. You can just assume this is the target. This is the drug. Okay, so both are complementary in shape, so that easily can be bind into the binding pocket on the active site of the target. Okay, so I think these things no need to discuss in things. What are diseases? I think you may be all well versed about that. Sometimes I used to take some lectures for the masters in chemistry students as well as the faculty. So that time so they may be required what is the disease, what is the classification of the disease, all those things they may be requiring. So that's the reason I just give this slide. Okay, so now what are the drugs and drug targets? So when you say the drug means, that it is a small molecule that will be binding, interact and modulate the activity of the biological receptors. That is the drug. And what are receptors? Receptors are the macromolecules. Okay, receptors are the proteins, the macromolecules that bind and interact with other molecules to perform the numerous functions required for the maintenance of the life. Okay, when there is any malfunction, so then we will be giving the drug to change the nature of the. Cannot change the nature to the signaling of the receptor, signaling of the macromolecule. We can change. So the macromolecule is considered as a lock. Okay. This is the macromolecule. So it is considered as a lock. And drugs are the small molecule. So it is considered as a key. So our duty is we have to find a particular key. To open or close the lock. As you all know that all keys cannot open or close the particular any all the locks, right? So only the particular keys can be open or lock the particular locks, right? So that is our aim is to find a particular find a appropriate key so that it can open the lock. So that is the very simple concept. So this is the lock and key mechanism. So I think this aspect also, I think you are familiar with these aspects. What are agonists and what are antagonists? In agonist binding with receptors, there will be a various cellular response will be there. Either it can be ion modulations will be there, or DNA transcription modulation. Suppose if it is an enzyme, so the agonist means the molecules are binding with the enzymes. Either it can be activate the enzyme or inhibit the activity of the enzyme. There are two things. Antagonist when it bind with receptors, there will not be any further signaling. That means that the effect will be blocked. So this is the ligand, agonist and antagonist. So here they are mentioning ligand. What does ligand means? It is a substance present endogenous. So endogenous ligand is the normal physiological function. Okay, agonist means it's a like external drug, which mimics the endogenous ligand. It will also produce a similar effect. And antagonist block the effect. So there will not be the effect when antagonist is binding with receptors. Okay. And how drugs are designed and discovered. So as I already told you, we have to find the drug target identification. That means the lock. So we should know the full structure of the lock. Okay, that means full details about the lock. Then only they will be able to design the key. Unless we know the details about the lock, we cannot design the key. So first we have to do thorough study for identifying the target, and it has to be validated. That particular receptors or particular enzyme really involved in the particular disease process. Then only he will be able to design the key for that. That is, design the drug for that. So, so from there they can get the key. So in the like public domain, there are so many molecules, chemical structures are available. So for example, in natural and synthetic chemical libraries, are billions of molecules, billions of chemical compounds will get it in the public domain. And new chemical entities, there will be thousands of new chemical entities will be there. And lead compounds also is there. For it will be like thousands. Okay, and clinical candidates. Okay, so that will be hundreds. And okay, finally the drugs, still hundreds. So what are the why I mentioned this thing is that already existing molecules, that maybe there are so many chemical compounds already existing in the public domains. Those molecules we can extract, we can use those structures. Okay, whether those structures can be utilized as a drug for the particular target. Not necessarily you have to design your own molecule. So we can take the existing
Thank you. கவரம் கொடுங்க. சின்ன கொடுங்க. கொடுங்க. கொடுங்க சார். சார். பேரண்ட் ஒருத்தவங்க கோபிநாத் பேரண்ட் என்னாச்சு ஸ்டக் ஆயிடுச்சு என்னடா இது சவுண்டே வரலன்னு பார்த்தேன்
இல்ல சார் அவர் சிக்னல் ப்ராப்ளம் போல அவர் போயிட்டார் சார் இல்ல சார்
சிக்னல் ப்ராப்ளம்
hello. Yeah, okay, I'm sorry. There was some disturbance in the network. Okay, fine.
Okay,
So
okay.
I'm just continuing. Yeah, I'm continuing.
சரி வாங்க ஒரு பர்சன் காமிக்கிறேன்
So from the public domain, so we have so many chemical structures are available. So those structures can be utilized to generate the lead molecules for the particular target. Okay, so those lead molecules can be a successful clinical candidates, and then can be a successful, ultimately that can be a successful drugs as well. Actually, this is a drug discovery cycle. You can say, okay, I can so this is the typical drug discovery cycle. Suppose if you have the 3D structure of the target at ligand, so based on the structure of the 3D structure of the target, we can design the new compounds. So how we can design the new compounds? So those molecules, it can be taken from the chemical space. That means from the public domains, there are so many chemical libraries are available. So from the chemical libraries, we can search those molecules based on the structure of the 3D structure of the 3-dimensional structure of the target. So from there they can, so first we take the
Sorry for interruption, sir. Slide
sir.
is not visible, sir.
Okay,
Presentation slide is not visible.
okay, just a minute.
Again, share your PPT, sir.
Yeah, okay, yes, I'm doing it.
நீங்க சேர்க்க வேண்டாமா சார்? எங்க அண்ணன் பட்டாமாஸ். இது யாரு? குழந்தை. இங்க வாங்க, எங்க தளபதி தெரியுதா? நீங்கதான் அப்படி
Yes, sir,
இளைய
now
தளபதி,
visible.
நீங்க தளபதி,
Is it visible now?
நீங்க தமாஷ்.
Yes, visible now, sir.
Okay, fine. Okay, so this is the typical drug discovery cycle.
நீங்க அவங்களுக்குலாம் அண்ணா மாதிரி.
So first you have to start from
Continue, sir. Yes,
Yeah,
yes,
okay.
continue, sir.
So this is the so we have to start from the 3D structure of the target. That means the protein molecules, RNAs and many things. So first in hand, so already we have the chemical 3D structure of the target. From the so using this we can design the new molecules. So how we can design the new molecules? We can obtain the new molecules from the chemical space. Chemical space means from the chemical libraries, online chemical libraries, so many chemical libraries are available. So from those chemical libraries, we can get structures. Okay? So that structure should be suitable for the particular target. Okay. So what we can do is initially we can have suppose for example hundreds of molecules. Okay. And then we can do the docking studies, in silico studies. So in silico studies we can filter it. So we can have the lead molecules from the docking studies, both pharmacodynamic and pharmacokinetics, as well as toxicity as well. We can do it only in the in silico studies, and then we can have your lead molecules from the in silico studies. So only lead molecules we can take it to the, so that is called as virtual hits. In silico studies, then we can have virtual kits with the lead molecules. Okay. Only those molecules we can go for synthesizing. For synthesizing, we can have SAR studies, QSR studies. We can go for it. And then if there is any like secondary assays are required or ADB screening, that is in the in vivo. Okay, that is preclinical studies. You can do it. Then it can be moved to the market. After getting approval, it can be moved to the market. So this is what I think you may be knowing this from starting. All people know. Pharmacy background, they will be knowing this drug Liptor. One of the it is very established molecule for lipid lowering drugs. Those are having high cholesterol. So atorvastatin, one of the drug. Most of the physicians they are prescribing this drug. So actually, these drugs were developed by the company Pfizer in the USA. So they developed the drug using this in silico studies. So these are very successful molecules reducing the cholesterol level. So actually they earned a lot of revenue from these molecules. Like 1996 to 2012, like $141 they generated using this. Actually, the patent was ended during 2010. So after that, after 2010, other companies also they are producing these molecules. Even nowadays, even generic product also it's available for starting. And this is another molecules, biological molecules. This is for the rheumatoid arthritis and psoriasis. They developed these molecules. Humira. This is also a successful molecules that was developed using the in silico studies. Okay, so then we'll move to the what are the applications of the computational tools in the drug target discoveries. So, in my previous slides, many times I was talking about the target, okay, the protein, receptor, enzymes. Okay, how to get those targets? How to get the details about the target? Either it can be receptor, it can be enzyme. Okay, how to get the exact. So we, at last, for doing the in silico studies, at last we need the three-dimensional structure of the target. Unless we get the three-dimensional structure of the target, we cannot go for in silico studies. So how to get? So we have various tools are there available. One is the single crystal X-ray diffraction techniques. That is one techniques by which we can generate the three-dimensional shape of the and then protein NMR. Protein NMR also will helpful to get the structure of the target molecule and 3D electron microscope. Mostly these three techniques are widely used, okay, to establish the structure of the protein molecule, the target molecule. Among these, okay, single crystal X-ray is the prominent one. But mostly the problem is you have to get the single crystals. Then only these techniques can be used. Single crystal X-ray diffraction. Sometimes, no. Suppose even though we have so many sophisticated tools are available, sometimes some of the proteins, okay, the structure we will not be able to get it. So in that case, we can go for homology modeling. What is homology modeling means? Okay, we can generate the structure of the unknown protein. It means unknown target with the known structure. That is called as homology modeling. It means generating the structure of the unknown protein from the known protein having similar structure. So that is called as homology modeling. By using these techniques, we can get the three-dimensional structure of the target. So I will explain one by one. So X-ray crystallography, that means single crystal X-ray crystallography. Main thing is that for that you have to go for crystallization. Protein crystallization. You have to get the crystal. Single crystal is very important for doing the X-ray studies. And then crystal mounting. So once we get the crystals of the protein, single crystal, using the X-ray, we can get the diffraction pattern, right, in various phases. So that will generate the electron density of the particular single crystal. So using the various software tools, we can generate the 3D structure of the proteins. So for generating the 3D structure, we need electron density. Okay. For getting the electron density, we need the single crystal of the target. So this is how we can get the three-dimensional shape. This is one technique, X-ray crystallography. Next one is NMR spectroscopy. Okay, NMR spectroscopy, if you say like 1D or 2D NMR, it may not help for getting the structure of the three-dimensional shape of the protein molecule, because we know that, okay, if you take any target like any proteins like receptor or enzymes, okay, it will contains a number of amino acids, right? For example, if you have suppose if you have a molecule containing the 50 proton, imagine how many peaks you will be getting in the NMR spectroscopy. It's very difficult to elucidate the structure if the number of protons are more. Okay, maybe this 1D and 2D NMR, it will be useful for small molecules. When you synthesize the molecules, or if you isolate some molecules from the natural product, that time you can use the 1D or 2D NMR, you can use it, because the number of protons will be less, right? But in the case of protein molecules, okay, there will be a number of protons will be there. So it is not possible 1D and 2D. So here you have to use the three-dimensional 3D NMR. You are supposed to use it. Using 3D NMR structure, 3D NMR spectrum, you can get the structure of the particular protein. And this is the homology modeling I have already told you. So this one, it will be useful when, okay, when suppose sometimes sometimes we will not be able to crystallize the protein. So that time you cannot use the you cannot use the X-ray techniques. You cannot use it. Sometimes NMR, 3D NMR also will not give the proper spectrum. So that times, okay, how to get the structure means by using homology modeling. Homology modeling means getting the structure from the known proteins. That is the homology modeling. Okay, so now I am coming to the so far I have discussed so many aspects about the drug discovery. So now I am moving to the next part, the target based virtual screening and ligand based virtual screening. So target based virtual screening is the, okay, you can call as structure based drug design, structure based drug discovery. That means in this what we'll be doing is the, okay, so we will be designing the molecules based on the information of the target. Based on the target, we will be developing our own molecules. That is called as target based virtual screening or structure based drug design. So the other aspect is ligand based virtual screening. Okay, so in this ligand based virtual screening means, okay, based on the knowledge of the existing drug molecules. Suppose in some cases, if you are not able to get the three-dimensional structure of the protein, okay, so in that case, okay, we can go for ligand based virtual screening. In ligand based virtual screening, they can develop the molecules based on the knowledge of the existing molecules for particular target. So these are the two aspects. One is target based virtual screening. In other next aspect is the ligand based virtual screening. Okay, so now I will just go one by one. First I will finish the target based virtual screening, then I will move to the ligand based virtual screening. Okay, so target based virtual screening, there are 6 major steps are there. One is the selection of the druggable target, and then selection of 3D geometry of target, and then we have to find the molecular flexibility and binding site parameters. So first we have to select the druggable target, and then after getting the target, we have to go for three-dimensional geometry of the target. Okay. So and then we can select the getting these after selecting the target, we can select the ligands. So ligands selection from the databases we can do either by FSR database, that means filtering, scoring, and ranking. So that is called as FSR. Okay. So first we selected the target, then selection of the ligands, then we can go for the virtual screening. Locking our molecule with the particular target. The final step is the analysis of docking results. So these are the 6 major steps that are involved in the target-based virtual screening. So I will just go one by one. Selection of druggable target. What is mean by selection of druggable target? So that means the identification of the target with therapeutic potential. That is most important. So you have to select the target having the therapeutic potential. And then once we identify, you have to evaluate it, the druggability, whether that's really the target is involved, responsible for the particular activity. So in this we can go for what are the amino acids are present in the particular target, what is the composition, size, shape, geometry, polarity, surfaces. Okay, that means entire details we need it. Then only we can able to design our molecule. So once we have the target, we should have the entire details about the particular target. and then selection of the 3D geometry of the target. This is also, then the next step is the first, once you get the target, then you should have the three-dimensional structure of the target. Unless if you have the three-dimensional structure of the target, you cannot design the molecules. So in this, I already mentioned that there are so many techniques are involved. One is the XRD and NMR. Mostly 3D NMR is used, and homology modeling also, it will be used to get the three-dimensional shape of the target. And then we have to see the molecular flexibility of the proteins, that means the target, the ligand, whether how flexible the target is, whether it is the structure is rigid or flexible. So there are various docking methods are there, like based on the rigidity of the both target as well as the drug. So we can design what kind of docking procedures we can adopt. So we can have like both the receptors and ligands are flexible, or only ligands are flexible. So all those details we should have in hand before docking. And number of degrees of freedom, degree of flexibility. So suppose, for example, I'm talking about the degree of flexibility. What is the degree of flexibility means? Suppose, for example, in simple terms, if you take a benzene ring, right, or it's a cyclohexane ring, and N-hexane, both the number of molecular formula or molecular weights are same, right? Molecular formula will be same. N-hexane as well as cyclohexane, right? Almost the same. But in case of N-hexane, it is a flexible molecule because it is carbon-carbon single bond, right? But in case of cyclohexane, it is a cyclic compound, it's a rigid molecule. So the number of conformations will be limited in the cyclohexane, whereas in the N-hexane, there are 6 carbons are there, right? So so each carbon will have its own flexibility. So degree of what is the degree of the flexibility? That's also should be taken into account. And the next one is the defining of binding site parameters. So what is the binding site? That binding site means what is the active site. So we have to define, before going for the docking, we have to define what is the binding site, what is the active site's geometry. So we have to give the x-axis value, y-axis value, and z-axis value. So all those values we have to give it. So, see, this is only getting the three-dimensional shape. 3D structure of the protein molecules will not help. See, from the whole protein structure, we have to see where exactly the binding site is. Unless if you suppose we fail in locating the binding site, your entire process will be, you will not get the fruitful result, right? So you have to define exactly where the binding site in the macromolecule, because the drug is not going to bind with the whole receptor or whole enzyme. The drug is going to bind only to the particular part of the target. So they have to define where is exactly the binding site, so that the drug can bind with that. So that we can have the various ligand receptor interactions. That means the drug receptor interactions. And then selection of ligand databases. So once you have so first thing is that you already selected the target, and then you define the binding site, that is the active site. Then you can select based on this knowledge, they can develop the drug molecules. So we have various, developing the drug molecules, we have various databases available. One is zinc database is there. And then you may be knowing, having a pharmacy background, you will be knowing various databases like zinc, and I can say drug bank is available. There are so many databases, right? So from there you can get the structures. Sometimes some chemical libraries are open source, some are commercial. So you have to pay for accessing the sites. You have to pay for accessing those structures. Most of the times, no, most of the structures it is open source. So okay, free of cost we can develop, we can download the structures. And sometimes we have API like databases, like active pharmaceutical ingredient like databases also are available. Not only chemical databases, API like databases, drug like databases are available. Okay. There are so many databases available in public domain. So from the databases we have to do the mining. That is our part. There are so many structures in the public domain. Okay. So based on our requirement, we have to select the molecule from the databases. We have to mine it. So how we can do it? By using the filtering, scoring, and ranking techniques. Okay, based on the prerequisite properties. Based on this, what is the prerequisite properties? Based on the structure of the three-dimensional shape of the target. Based on this, we have to mine the databases so that they can get a good, okay, what do you call that? Good molecules, okay, from the databases. So this is how, okay, actually the docking takes place. The next step is the docking. Next step in the TBVS is the docking of the selected FSR ligands, filtering, scoring, and ranking. So now we have the two parts we already established. One is the target, and next one is we have the drug. So two things are there we have in our hand. The next part is that you have to dock it and and analyze the docking results. So using the previous all the techniques, okay, whatever I have explained. So we have developed here the 3D structure of the target in one hand. In another hand, I have the drug molecules, number of drug molecules. The target will be one, right? But the drug molecules will be, number of drug molecules will be there. So I will be keep binding those drug molecules, each drug molecule to a particular target, and I will see which drug molecule having a good docking score. Okay, that means docking score in the sense good binding affinity. So, okay, based on the binding affinity, that is the dock score. So I have to select those structures, those drug chemical molecules. So then those chemical molecules I can go for synthesizing. So this is how, okay, a drug design is happening. So this is one. In one hand, you have a receptor. The receptor will be one receptor will be there. Okay, and then you have multiple ligands will be there. Multiple molecules will be there. Okay, each ligand do the docking. Each ligand do the docking and see which ligand showing the good affinity for a particular target. Select only those ligands. Suppose we have like hundreds of ligands. Okay, you can just dock each ligand. Okay, either you can all 100 molecules you can. So we have good algorithms are available. Software algorithms are available. Okay, either you can dock individual molecules, you can dock it, or all 100 molecules you can dock together also.
Sir.
But all 100 molecules you will get the docking score separately. Right? So based on the docking score, we can select only those molecules which is having good binding affinity. Only those molecules we can select it and you can go for further studies. That means for synthesizing the molecules. For docking, we have a lot of softwares. Okay, a lot of online tools are available. The very prominent one is the Autodock, because Autodock is the worldwide. Okay, most of the people are using the Autodock, and most you can say this is very user friendly, and it is available free of cost. User friendly as well as free of cost, and some tools like Gold, Glider, all those are all commercial. You have to pay for the particular tools, and you can use the particular softwares for docking. But Autodock is so far is freely available. So these are the various docking tools are available. This is the docking parameters. Docking parameters means One is I already told you. Okay, what is the binding site? It is package specific parameters, grid parameters. Grid parameters in the sense it is what is the active site parameters. So you have to define x-axis, y-axis, and z-axis. So you have that will form a grid. So in that grid only the new molecule is going to bind, and it will give the binding score. And flexible residues, a number of runs, okay, how many runs you are doing. So that are all docking parameters. And docking post generation. After docking is finalized, you will get the you will get a complex. That means that means the target that is bound with the our ligand. Okay, so from the docking post, after docking post generation, you can see that what is the docking score, as well as what is the interaction, what are the types of interaction that is taking place with the target and the drug. Like as you may be knowing that there are so many drug receptor interaction. For example, the most prominent one is the hydrogen bonding, hydrophobic, electrostatic, right? So there are, so what are the actual drug receptor is taking place. So we can visualize the things. So there are mostly there will be three types of blockings there. Theoretically, one is the rigid docking, flexible docking, induced fit docking. In rigid docking, okay, the target as well as the drug both are rigid. Okay, in the docking, both will just bind. It is called as rigid docking. In flexible docking, what happened is when in flexible docking, what happened? Okay, this the structure is okay. The structure of the target is rigid. Okay, when the molecule when the molecule is approaching the target, so there will be a conformational changes in the drug. After conformational changes, so the drug will bind with the target. So it's called as flexible docking. So here the molecules, you can see the shape of the drug here and shape of the drug here. So the molecule is flexible, right? So that it is changing its structure based on the shape of the target. Here, neither the target structure nor the drug structure are complementary in shape. So both cannot be bind. In this case, what happened is, okay. So suppose it like this. So both cannot bind properly. This is the structure, and this is the your drug. So when both are coming together, what happened? There will be conformational changes. There will be conformational changes when both are coming together. Okay, so that it will bind it. So it is called as induced fit docking. Induced fit docking means okay. The what do you call that? The structure of the target. That means the receptor inducing the conformational changes in the drug. At the same time, the drug also induces the conformational changes in the protein, the target. So, okay, both will induce the conformational changes. So even though both are not complementary in shape, okay, but during docking, there will be the flexibility will be induced so that both will bind. So it is called as induced fit docking. And finally is the analysis of the docked process. What are the interaction points? Interaction point means okay, what is the various what do you say drug receptor interaction? What are the various drug receptor interaction? Okay, and then what is the binding score? Binding or docking score? So this is the docking score. It is we will be getting in the what is the energy? Okay, energy of the complex, the drug receptor complex. So we know that in basic chemistry we know that least binding energy having the most stable structure, right? So after docking also we will be getting energy of the molecule, energy of the complex. Okay, so we can find out which is having least binding energy. So those molecule having is having good binding affinity. So then that way we can come to a conclusion. and then you can get the optimization of the binding interaction, pharmacophoric percentages. What are the pharmacophoric percentages? So that one I'll explain in the coming slides. Of course, okay. So these are all the target-based visual screening. Of course, there are some limitations of this target-based visual screening. What are the limitations? Is the poor quality of the 3D protein molecules. Suppose if you're not the 3D structure of the target molecule is not proper, okay, then your entire docking procedure will be no use. You will not getting the prominent results. Whatever results you are getting, that will not be a real one. And sometimes know what algorithms are using, because each tools, each software, they're using different algorithms. Okay. So based on the algorithms also, you will get the ambiguous results you will be getting. Right? So those are some of the limitations in the target-based visual screening. So now I will move to these are some of the molecules that were developed using structure-based drug design or target-based visual screening. Starting from the captopril, there are various anti-HIV and antiviral drugs have been developed using this structure-based drug design. So with this, I'll move to the last part of the today's session, ligand-based drug design, or you can say ligand-based visual screening. So far what we have done is, okay, based on the structure of the target, we design the molecules. Now in this approach, what we can do is, so suppose we don't have the structure of the three-dimensional shape, we don't have the enough details about the target. So in that case, what we can do is, okay, based on the knowledge of the based on the details of the existing drugs, we can develop our new molecules. That is called as ligand-based visual screening or ligand-based drug design. So mostly this will be in these are ligands. Mostly will be in small organic molecules. So those should be in complementary in shape to the target. This should be oppositely charged. It means this should be complementary in shape as well as the charge. So that will the ligand will interact with the target, bind to the target, and it can activate or inhibit the biomolecules. Okay. So in this, in the target-based visual screening, or ligand-based drug design, so we are relying on the pharmacophore. Okay, as I already told you, based on the knowledge of the existing drug. Okay, so based on the knowledge, what does the knowledge means? Based on the details. The details is the pharmacophore. Based on the pharmacophore, because once we have the pharmacophore, okay, so we can develop our own molecules. As you all, if you're a pharmacy background, you know what is pharmacophore, right? The pharmacophore is the essential part of the drug, which are responsible for the activity, right? That means essential part of the drug, which is going to bind to the target. So that is the pharmacophore. So in this we have here 1D pharmacophore, 2D pharmacophore, and 3D pharmacophore. 1D pharmacophore, that means it gives only what are the groups, what are the parts of the molecules that is responsible for the activity. That is called as 1D pharmacophore. That is how many aryl sites are there, how many hydrogen bond donors are there, how many hydrogen bond acceptors. Okay, so like that. That's only those informations. So suppose antihistamine means you should have 2 aryl groups will be there, okay, and then 1 basic amino groups will be there, like that. So that is called as 1D pharmacophore. In 2D pharmacophore, and what is the relative positions between these groups? That is called as 2D pharmacophore. In 3D pharmacophore, it is the geometry between the groups or the atoms, the molecules. That is called as 3D pharmacophore. So 3D pharmacophore actually gives the exact picture. Okay, based on 3D pharmacophore, we can design the new molecules. So this is the typical pharmacophoric model, right? So you can see there is a aryl group is there. Okay, there is another group, and then there is a hydrogen bond donor, hydrogen bond acceptors. Okay, and what is the relative position is between each groups? And you can say, so I just explained this pharmacophore using the a well-known molecule, morphine. Morphine is a opioid analgesic molecules. Okay. So this is the opioid morphine structure. I think you are all studied about the structure of the morphine structure. Still, I remember. Yeah, I think we studied like in chemistry of natural product. So that we have studied the morphine structures. So I'm just taking as an example to explain the how we can develop the pharmacophoric model from the existing molecule. This is the pharmacophore, structure of the morphine. So in this morphine, you can see what are the groups are responsible for the activity. So morphine, we know that it is an opioid analgesic, right? So what are the groups are responsible for? The entire molecules are not responsible. Okay? Any drug molecules, all molecules will not, all part of the molecule will not going to bind with the receptor. Only the part of the molecules, only essential part of the molecule is going to bind with the receptor. So here, what are the essential part is the aryl group and OH. That is phenolic hydroxyl group, and then this aryl, and then this tertiary amino group. So this is all the important for the opioid analgesic. Not the other structures, other part of the molecules are not responsible for the activity or binding with receptor. So now, so what are the information we got for the morphine? One aryl group, phenolic OH group, and tertiary amine. So this is the this is the information we got. Now, how this is attached? Of course, the OH is directly attached to the phenyl group, and then nitrogen is attached to this aromatic ring through 3 carbon system. 1, 2, 3. 3 carbon system is attached to the aryl group. So from this, we have derived a final pharmacophoric structure. So these are the exact part of the molecule, morphine molecule, responsible for the activity. You can see this. Okay. So from here, we have come to a pharmacophoric. This is the pharmacophoric model for the opioid. So with this information, they can develop the molecules. Okay. So this is the molecules. Morphine molecules. So they have developed the metazosine and levonorphenol. So in metazosine, levonorphenol, the structure is different. You can see the structure. All 3 structures are different. But what is it? What is unique? What is common in all structures is the pharmacophoric model is same. It is intact. In this, you see this. Okay. In this also, phenolic OH group, aryl group, and tertiary amino group. These both are attached to 3 carbon system. Here also, so the pharmacophoric model is intact. So this is how you can see this. Okay. So this is the good representation of the pharmacophoric model. So this is morphine, metazosine, levonorphenol. Right? All are belongs to the same category because the pharmacophoric is intact. So based on this, for each what you call drug molecules. Suppose we have a series of molecules, you can develop your own pharmacophoric model. And we can, with the use of the pharmacophoric model, we can develop our own new drug, new chemical entities. We can develop it. So the pharmacophore is very, very, very important. What it means is for a desirable binding interaction. So how it is used? This is the pharmacophoric, useful for one to prepare a more active molecules. So using the pharmacophoric models, we can avoid the unnecessarily part of the molecules. So we can incorporate only the essential part which are required. That means pharmacophoric part in the molecules. So we can remove the excessive groups, excessive part in a molecule, so that we can reduce the toxicity and cost of the production. So this can be done through the synthetic modification. how to do the synthetic modification? Means the ROH, or for example, if a molecule contain ROH, so this can be converted to ROCH3. Okay, so we can see whether the OH group is important or not in the molecule. So ROH is converting to ROCH3. So in this, the hydrogen is replaced by CH3 group. So in this case, when ROH is converted to ROCH3, okay, we can come to conclusion whether this ROH is important or not. We can see the activity of the only ROH, one molecule, ROCH3, another molecules. Okay, so we can compare both the activity. So then we can come to a conclusion whether this ROCH3 is required or ROH is required for the activity for interaction. Similarly, suppose RNH2, so it can be converted to amide. Then we can say whether both the hydrogens are important or not. So 3D pharmacophore, what is 3D pharmacophore? Is the three-dimensional pharmacophore, specify the spatial relationship between the groups. Okay, and also it defines what is the angle between the two groups, and what is the dihedral angle between three groups. Dihedral angles usually we measure from three different groups, right? So this is the commonly used. Okay, 3D pharmacophore antihistamides. It contains the two aromatic rings, right, and one tertiary nitrogen atom. Okay, that is attached in a specified manner, but because this distance between this aromatic and this aromatic. So this aromatic can be a phenyl ring, or it can be a heteroaromatic ring also. Okay, either pyridine also it can be used here, but it should be aromatic. So two aromatic ring. So what should be the positions? So it should be like 4.7 to 4.1 angstrom. So distance between these two things, between these two parts should be this should be within this range. And this center of the aromatic ring to the tertiary nitrogen atom within this region. And this aromatic to this nitrogen within this distance. So this is the typical pharmacophoric of antihistaminic agents. And you can see this is again I'm just coming into the morphine molecule, just representing. You can see this, okay, aromatic ring. In the morphine you can see this is aromatic, and this is the phenolic OH group, electronegative atom, and this is the tertiary nitrogen atom, and this is the how it is attached. So this I already mentioned that in previous one. Okay, what is the distance, right? The distance is important, not only the groups, right? What are the distance? So this distance are also important. This you can mention here. Okay, what is the distance between this center of the aromatic ring to this phenolic OH group? Okay, and the angle also is important. What is the angle between this one? What is the angle between this one? Okay, so this is the typical pharmacophoric model. So that we have obtained from the morphine structure. So in my previous slides, I am mentioning about the conformation, the flexible molecules, rigid molecules. Maybe I'm just finishing in, okay. Mostly in 5 minutes I will be finishing. I know that I'm running out of time. Please bear with me. I will just finishing mostly in 5 minutes. So in the previous slides, I was mentioning about the flexible molecule and rigid molecules. Okay, flexible molecules having the number of conformations to be more. So when number of conformations are more, what happens is each conformation will binding with the different receptors. Each conformation will binding with the different receptors. During which what happens is, okay, so when it's binding with the different receptors, okay, so the selectivity is not there. So because of lack of selectivity, so you may expect the toxicity, right? So when a drug is binding with the multiple receptors, so obviously we can expect that what is the toxicity, right? So in that case, okay, rigid molecules, mostly it will be, okay, the number of conformations will be restricted. So it will going to bind with the only one type of receptors. So the toxicity will be minimized. For example, the one classical example I have taken here is the dopamine. So this is the structure of the dopamine. Okay, you can see there is a carbon-carbon single bonds are there. During those of which, okay, it will be having a number of conformation. So in the another structure, okay, the pharmacophore is same because whatever the structure is existing, we have not changed the structure. But okay, this carbon-carbon single bond, it has been confined in a cyclic structure, because of which the number of conformation is restricted. What do you call that? Rotation of the molecules will be restricted, right? You can see this is the flexible molecule, and the same molecule, same, but it is restricted because this amino group attached to the phenyl ring using 2 carbon system. Here also 2 carbon, 2 carbon system. And another molecule, you can see this here also. Okay, the structure seems to be different. Okay, but okay, it will be same. The pharmacophore is same. Phenyl ring, amino group attached to both are attached to 2 carbon system. So these two molecules, okay, it is considered as a rigid molecule. So when it's a rigid molecule is there, the number of conformations will be reduced. So when the number of conformations is reduced, okay, so we can expect the toxicity will be minimal. No toxicity will be minimal. When there is number of conformations are more, so it will going to bind with the multiple receptors. So we have, okay, toxicity will be more. So this is the pharmacophores from target binding site. So for example, suppose I told you the target like pharmacophore. How this pharmacophore is going to bind, how it will be useful. Suppose if you take this, this is the basic or positive center here. Okay, one side we have aromatic center here, and here we have a hydrogen bond donor. This is the pharmacophore, okay, a particular class of drugs. How it is going to bind the receptors? Suppose if we have a positive, basic or positive center, so that is okay. So that will have a positive charge here in the molecule. So that is going to bind with the negative charge in the target. So in the target we have the aspartate. Aspartate, we know that aspartate amino acid, we have carboxylic acid, right? So it will carry the negative charge. So that will be so here the molecule we have positive charge. So both are going to bind through electrostatic interaction. But in this case we have hydrogen bond donor acceptor. So here we have hydrogen bond donor acceptor. So that is going to bind with the serine molecule. Serine molecule contain OH group. Here we have hydrogen bond donor. Excuse me. Here we have hydrogen bond donor acceptor. So here in this part of the molecule is going to bind with the serine present in the target. So by hydrogen bonding interaction. And then here in the target we have phenyl. Okay, in the pharmacophore we have aromatic center. So both will interact through the hydrophobic interaction. So this is how, okay, yeah. So when you say pharmacophore, means this pharmacophore is responsible for the interaction with the receptor. Okay, so this part, okay, is responsible for the electrostatic interaction with the aspartate, and this part is responsible for the hydrogen bonding interaction with the serine. And this part, aromatic center, responsible for the hydrophobic interaction with the phenylalanine in the target. Okay, so this is about the pharmacophore. I'm just moving to the next one. Ligand-based drug design. What are the methods we can adapt? One is the virtual screening. So virtual screening in the sense, okay, what do you call that? So generate a number of structure. That means in silico method generate a number of structures. That means either you can download the structures from the database, okay, and then okay, bind with the particular target. That is virtual screening, you can say. And then de novo design. So de novo design means once if you have a pharmacophoric model, pharmacophoric design, okay, so what are the requirements? So each one, okay, so we can design. Okay, so this part we need a basic center, and here we need a hydrogen bond acceptor or donor. And another part we should have a like a phenyl ring. Okay, so So we can have what kind of group we can have for hydrogen bonding. So what kind of groups we can have for hydrophobic interaction. So piece by piece, you take one piece from here, another piece, or this piece. All we together you just make it. So de novo design. De novo design means anything from scratch. So that is called as de novo design. And then optimization of the known ligands. Suppose we already existing drugs are there. Okay, so we can optimize that structures. So based on the requirement, we can optimize it. So now we have done the structure-based drug design and ligand-based drug design. And the final part will be docking. Okay, so docking is the exactly in simple words, but what is the docking is the just finding the right key for the lock. That's the docking. Okay, I'm just skipping some of the things. So finally, I'll just I'll show you. So okay, the next one is the stages of the docking. What are the stages of the docking? One is the first, as I already mentioned also, either you have to first thing is that selection of the target, the receptor selection, and then you have to prepare the docking. Ligand selection and preparation. Okay, ligand in sense the drug, whatever the molecule you are doing. Ligand selection and preparation, and then do the docking, and then evaluating the results. So this is the actual schematic diagram. Okay, first target selection, target preparation. Ligand selection, ligand preparation. So now we have prepared the target, we have prepared the ligand. So then both we can do for the docking. Once docking is completed, we can go for evaluating the results. That means in evaluating result means mainly two output you will be getting in any docking program. One is the docking score, that means the binding energy. What is the binding energy? The next one is what are the various interactions. So these both informations you will get it. You have to analyze it. Okay. So these are the various steps that are involved in the docking program. But I think this one is required a separate what do you call that? Separate session. Maybe in future. Okay. If time permits, okay. If we are organizing any programs, maybe I can give you one demo class also, how to do the docking also, using how to dock. Why not? Okay. So the applications of docking, okay. I no need to mention much about the applications of docking. Applications of docking is docking will minimize our time, okay, our resources, our cost, okay, everything will be minimized. That is main things. Before going to the lab, using docking, we can have a idea about the drug, how it is going to interact. What about the pharmacokinetic and dynamic, as well as toxicity also you can predict it. To some extent, we can predict toxicity also using in silico studies. Okay, and thanks for all of you for your patience. I think I have taken more time. I think 1.5 hours I have taken. Now I am okay. For any query or any suggestions or any doubts, I'm happy to address.
Thank you, sir, for your wonderful presentation and elaborative presentations on the drug discovery as well as docking studies, and especially the tools what you have used in the nowadays for the research facilities. That will be one of the enthusiastic session. Even what we have discussed, we know about the very good knowledge on the applications of computational tools in the drug discovery methods, structure-based drug designs, ligand-based drug designs, and the various stages as well as steps in drug docking studies. We give the much information about the young pharmacists as well as buddies and the present in the faculties. As well as researchers, they got the good knowledge about that area. And any doubts and anything need to discuss, I request the participant to unmute your mic and you can ask. Sir, Dean sir, your mic is not switched on, I think so.
Venkatesh Perumal. So thank you very much. You have given an excellent speech. It is very much excellent. I am very much happy. So, and we are in the same field, that is pharmaceutical chemistry. Some of my scholars, they are also working in docking.
Okay.
So definitely next session, so we might be having either in person also in our campus,
Yeah.
a workshop on dogging. So thank you very much.
Okay. So
And I thank all.
suppose
Solva.
yeah, if you invite, maybe I will be fly from Saudi Arabia to Chennai. I can
No, no,
maybe I
no.
can have one. Yeah.
When you are
No, no,
in
yeah, of
Chennai
course.
or when you
Yes,
are
yes,
in
yes,
India,
yes, of
we will
course.
call you.
Yes, of course, of course. Okay, when I'm coming for vacation, maybe I can visit
Yeah,
you.
yeah, yeah,
Of
yeah.
course.
Already we
Of
missed
course, I
you in
have to be.
vacation.
Yes, yes, of course. I have to visit. I have to visit your campus, maybe for visiting my old friend. Two friends are there. Okay.
Thank you.
So I have to visit. Maybe during visit, maybe we can have one session also.
Yes,
So for personal interaction.
definitely.
Yes.
And I thank my faculty members, everybody. So particularly Jairam Kani madam, Saravanan sir, and all the members who have taken active participation. So Helen madam, of course. So they have made an active participation designing the browser and then day by day calling the people. So day by day, all the faculty members, the fresh people, they have joined as an invitee. So calling about the invitee sessions. So thank you very much. All these 5 days, this week went like anything for us. So it's a special week for us. Thank you once again for all my faculty members who made our conference, that is FDP, grand success. Thank you.
I request Jairam Kani madam to share your few things about this FDP.
Good afternoon, Perumal.
Yeah. Yes, madam.
Thank you for sharing your knowledge with us. So we are happy to see you as a resource person here. So during college days, we have seen you as a sincere, hard worker, quiet and studious person. Today you have proved that. Today you have proved by your presentation. It was really a great one. It showed your deep knowledge in molecular talking. Also, our students also enjoyed the session. So again, thank you for spending your valuable time with us. We will meet again.
Okay, okay.
Great.
Thanks for your
Thank you.
word.
Thank you. Thank you.
Now I request Lata madam to give the closing session speech and followed by a vote of thanks.
Vote of thanks. As we come to close of this enriching faculty development program, I take this opportunity to extend a heartfelt gratitude to everyone who made this event a success. Firstly, our deepest thanks to Dr. R. Venkatesha Perumal sir for his invaluable contribution. Your extensive knowledge and insights in the fields of in silico molecular docking, pharmacokinetic studies, and DSP studies have been incredibly enlightening. We are grateful for your time and effort in sharing your expertise with us. We also thank Qasem University, Saudi Arabia, for their support in allowing Dr. R. Venkatesha Perumal sir to be a part of our program. A special thanks to all our participants for their active involvement and engagement throughout the sessions. Your enthusiasm and curiosity have made this program truly interactive and successful. Lastly, we extend our appreciation to the organizing committee and support staff for their seamless coordination and dedication, ensuring the smooth running of this program. Thank you all once again for making this faculty development program a memorable and a fruitful.
So thank you.
Thank you, ma'am. And once again, this is the time to thank all our resources persons for the various sessions after we start this FDP. Because each and every person, even in their busy schedule, they are accepted to give as a resource person to deliver a topic in this FDP. And they be a part of persons there is online 5 days faculty development program organized by our Faculty of Pharmacy by her on the advancement in the research and innovation in the pharmacy. And in this regard, they share their knowledge, share their feelings, and the various, we covered almost the various disciplines like drug discovery and the nanomaterial production and network pharmacology and pharmacogenomics and pharmacovigilance, even the industrial collaboration, nanoparticle as colloidal drug delivery systems, as well as the structure-based, ligand-based approaches in the drug discovery. So I thank the resource persons, Dr. Ashok Kumar Janagiraman, Assistant Professor, Kuala Lumpur, Malaysia, to give the speech about the recent advances in the metallic nanomaterials for the cancer therapies. Dr. Sheehan Prashanta, he gave the idea about the network pharmacology and pharmacogenomics approaches in the drug discovery. Dr. Mr. Ake Alim Shah, he gave the importance about the pharmacovigilance and current scenario from the TCS. Also, there are Dr. J. Ramesh Babu sir from Chibrolu Hanumaya Institute of Pharmaceutical Science, AP, gave a wonderful speech about the nanoparticle as a collateral drug delivery system and their basic approaches. Also, finally, Dr. R. Venkatesh Perumal sir, professor from Qasim University in Saudi Arabia, gave a wonderful speech about the innovative strategies in the drug discovery and exploring the structure-based and the ligand-based approaches. And this is the time to thank our organizing committees, our Chief Class Chairman, Dr. S. Jagatputhikan sir, our President, Dr. J. Sandeep Anand sir, our MD Madam, Dr. S. Sweta Sandeep Anand Madam, as well as the various patrons like Pro Chancellor, Dr. K. Vijay Bhaskar Raju sir, and Vice Chancellor, Sundarajan sir. And also our special address to Dr. Hari Prakash sir, and our most and important, the convener, Dr. R. Srinivasan sir, Dean Pharmacy, as well as the co-conveners, Dr. R. Saravanan and Dr. K. Rajaganapati sir. Also the coordinators, Mrs. N. Jairamakani madam, as well as Dr. W. Helen, and along with our various network teams and as well as our staff members, Dr. K. Rajaganapati sir, Vijay Lakshmi madam, Vasant Kumar sir, Jairamakani madam, Ilavarasi, Kalaivanan sir, Rakshana madam, and Hari Nandini, Vignesh, Rajita ma'am, Lata ma'am, and all the faculty members, they in and around this faculty development program to make it a grand success. Even for the online, even previously we organized in the offline manner only, but now we are initiated in the online for the FDP because we organize the various webinars in the online platform, and this is the first time we organized the FDP in online platform. It is a wonderful experience, even our staff members as well as the faculties. Really, I thank all the members in and around this particular bar. Once again, I thank all the faculties for this wonderful session to make a grand success. Thank you, sir.
Thank you, Gurmal. Thank you, sir. Thank you, Saravanan sir. Thank you, Jairam
Thank you.
madam.
Thank you very much, sir.
Thank you, sir.
Thank you.
Thank you. We can close the session.
Yes.